Manor works at the Dana-Farber, and he's also at the Hebrew University. And Manor, as you know, that mass spectrometry data is very complex, and it has many different dimensions. And it's usually quite problematic to analyze it thoroughly and in an easy way. And Manor is going to talk about several different strategies that he has developed on how to make looking at mass spectrometry data much easier and much quicker. Manor.
Put that here. Right. So the Dana-Farber, the Harvard Medical School, that's my professional hangout, and the Hebrew University is sort of my academic hangout. I'm apologizing for the large number of logos. I think this is the maximum there will ever be. I'm going to try and stick to one affiliation in future talks, but well, at the very least, I'm funded by all of you. So here we go. Mass-informatics is, the premise of this talk is that mass-informatics is a topic that deserves its name in the same way that there's bioinformatics or, it's clinical informatics. And I'm going to try and show that since there's a diversity of axes that at the center of which are spectra, it deserves its own topic. But a side effect of doing that is that I won't be able to cover all of these. So, but I will try, and so this thing will get a little scattered really quickly. So basically, the premise is that when you're looking at spectra with a mass spectrometer in the life sciences, you're doing this for a multitude of reasons. You might be interested in the technology itself. You might be interested in the biology that it generates. You might be interested to do quantitative work, or you might still be focusing primarily on ID, whether it's for small molecules or peptides. So there's a large diversity of things you could be doing, but at the center of these is always spectra. And that's the message, if there is any here, is that when you're looking at spectra from the perspective of doing all of these things, you get a slightly different computational flavor. And I'll try and show some examples of that. And since this is a very tool-oriented talk, so we're going to do two things. First is we're going to try to do some lab demos, which is certain to fail, especially since it's being recorded. And I'll start with one so that you, whatever happens, I think this one is a little tool that doesn't require a lot of setup, and it gives you something to walk away with. So it happens a lot that you have a peptide and you would want to just make sure where it shows up in the proteome. Sometimes you want to know where it shows up in the entire proteome. So you look up the peptide. I just want to show you, this will recur in the talk where you have a website and it has what's called a RESTful interface, so that it should be very self-explanatory. You go to here, you say, which database am I looking at and what is the peptide I'm interested in, right? So you could ask, how many times does France show up in the known proteome? Everybody does their name, etc. So basically, when you have a peptide and you want to know, not just in your organism, but just in all taxa, whether this peptide show up, this is a fast way to do it. And I just want to show you that, so it's an index, basically. And I'm just showing you here all the places in all species where the peptide Finland shows up. I just want to point out that there's an index that does not distinguish between isoleucine and leucine, which is a mass spec weirdness in that they have the same mass. And you'll see that that list becomes much larger because this is we're now looking up the occurrences of Finland or FL, NI, AND, etc. So the list is much longer. So if you ever have a peptide you want to look up what it is, or if you have a program that needs to look up, this is just pointing out that the same thing can generate just text. So you can have a program make this call and just get out all of the uniref locations. This is just a little useful web service. One final note about that, it's not just for looking up your name. The real use, if you will, is when you're doing metaproteomics experiments. So when you're looking at mixtures that have peptides that truly are coming from multiple species, and that's described in the relevant publication, which I'll add all of the relevant references when we post this. But basically, this is a simple, self-contained tool, which I like to start with because whatever happens, that's a very easy one to use. Okay, so and it's part of this mass informatics set of tools in the sense that you have a peptide. For example, this weirdness of not knowing whether it's a leucine or an isoleucine is a side effect of doing mass spectrometry. You don't get that when you sequence. Okay, so in order to demonstrate some more of these tools, we're going to need to look at a biological experiment. And so I'm going to start by setting up one of these experiments. And unfortunately, to show off a broad set of these tools, it's quite a complex experiment, both biochemically and in terms of how the experiment is run in the instrument. So just to calibrate, how much of the audience knows the distinction, say, between an MS1 and an MS2? Are you all comfortable? Yeah, okay. So what I'm really tempted to do the sort of mass spec refresher, which I always have ready, going over, really back to the basics. So I'm probably not going to be able to do it right now, but if you're interested afterwards, I have the slides for it. It explains a lot of this stuff, and I'd be happy to go over it with you. So I just now know that I'm losing half the people about on slide 5. So, but this is, if you want to afterwards, I'm happy to go over this. So let's set up the experiment. We're going to be looking at phosphoproteomics. So we're going to be measuring the intensity of occurrence of phosphotyrosines as we look at the mutants versus wild type. So we're going to look at some receptor, FLT3 receptor, which is going to respond to its ligand and phosphorylate some tyrosines and cause a whole signaling cascade. We're going to be looking at just the tyrosine component of it. So right there's a level of complexity in the proteomics in that we are not looking at the amount of entire peptides changing, we're looking at just the phosphosites. And what complicates this is that sometimes individual phosphosites will go in different directions on the same protein. So that's a level of complications that we're going to have to keep track of. We're going to be looking at the wild type without ligand, the wild type with ligand, and then 2 mutations that cause constitutive activation and are clinically relevant of this signaling cascade. So one of them is called the ITD mutation, and the other one is a point mutation called D835Y. They occur, they impact, they have a clinical impact, and so we would like to study the differences in how they affect the signaling cascades. Okay, so and we're going to try and get as many phosphosites as we can. Again, looking at phosphotyrosines. So let me show you how that's going to be done experimentally. We're going to take these 4 conditions, wild type with ligand, and then the 2 mutations. We're going to label them after digesting 2 peptides. We're going to enrich with antibodies for phosphotyrosines, do another follow-up enrichment just for general phospho, and then into the instrument with some pretty intense separation. The main point here is that we're able to measure 4 conditions at once, thanks to this iTrack label. Now, you may have heard about isotopic labeling in the past. The typical way that works is you have, say, 2 samples, and one of which is labeled heavy. And this is why I was asking about the distinction between MS1 and MS2. At the MS1 level, when you're surveying your peptides, you will see a heavy version and a light version, and the ratio will give you relative amounts. iTrack doesn't work that way because the label is engineered to have the same, all of the 4 variants of the label are engineered to have exactly the same mass. So you don't distinguish the source of the peptide in the various samples at the MS1 level. You do a tandem mass spec at MS2, and what happens is, in addition to the ID component of the spectrum, at the lowest end there will be 4 reporter ions representing the relative amounts of this peptide in the 4 conditions that you're studying, which in our case will be wild type, with ligand, one mutation, the other mutation. So in this case, this phosphosite responds equally to these 2 different mutations in the FLT3 receptor. Okay, so I'm just setting this up. This label has the nice property that in certain kinds of instruments you get all of the, and depending on how you run it, you get all of the information, both quantitative and qualitative, in one scan. And so just, again, to complicate things a little bit, we're going to run it differently. We're going to do 2 scans, one scan for ID, and then another scan optimized to get the quantitative information. If you're not doing mass spectrometry on a daily basis, it's not that important. I'm pointing it out because I want to show you some tools later on which allow you to dig into the data, and I want to prove to you that you can go into the minutest data level that you want, including this distinction. So we'll be able to see both of those scans. Okay. So that's the only reason I'm mentioning this. So the goal, we're going to be looking at a few algorithms and tools. The first challenge we're going to be looking at is a quantitative challenge where we're trying to get a sense of which of these ratios, now we're comparing these 3 conditions to the wild type, which of these ratios are statistically significant. You know, you've seen a lot of talks in the microarray realm, in the large next generation sequencing, where people are asking similar questions. When you're doing your typical proteomics, and you have multiple peptides that are assumed to be coming from the same protein, and you just care about protein amount, you can have some redundancy in some statistics because you're looking at multiple peptides. But like I mentioned earlier, these peptides from the same protein are legitimately going in different directions in terms of their response to the signaling cascade, to the mutation status. So we need to develop an error model which will allow you to make some kind of statement about individual scans. So that's the quantitative challenge here, which I'm going to demonstrate first. And so let me just show you first the raw data. These are all of the phosphosites that were characterized. You can see what is even apparent with a simple Western, that the D835Y causes a massive amount of upregulation in phosphotyrosine, ITD less, and just the ligand a whole lot less, especially since, and the biologists tell me to say this isn't serum starved. So there's already a lot going on in the background. We're just adding the ligand. One aspect of this, oftentimes algorithms that try and deal with this question of ratios and which are significant assume that most of the data isn't changing. So you'll get a bunch of stuff in the middle, and you'll use that to learn inherently where the limitations are of statistical significance. But here we can't do this because we're looking at a very selective subset of the proteome and peptide space, and it can legitimately move in its entirety. So this is, again, an avenue that's close to us, and we really need to reason on an individual scan level. Okay. So the natural thing to do in this situation is to first try and gather some statistics when there is no change. So this is sort of a null experiment where we labeled the same thing with 2 different labels, and we looked at the kind of ratios that we're seeing on a log scale, and we actually put on the x-axis the intensity, because this matters a lot in this kind of experiment. Basically, the more intense the peaks are, the more accurate the ratios can be. So this relationship is very well understood in the microarray arena again, and we wanted to take a look at that in our field and come up with some sort of variance function to link these two. The reason you want to do that is that if you sort of put this on its side and basically look at the ratios and ignore intensity, they will be coming from a distribution that's over-dispersed. It's basically not normal. So on a QQ plot with a normal distribution, the tails are heavy, and that's because some of these are very low in their variance and some are high, and they're just different distributions. And if you look at specific intensity ranges and look only in those, then the log ratios look normalish. And so what we did, and again, the details are in the publication, which will be attached with what we post. We fit a variance function, a continuous variance function, which then allows us to see where significance is in a way that's intensity dependent. So what that means in practice is that you're not needing to, in principle, just fix some threshold like twofold or half, which is often done even in literature. For what is significant, you can actually do it in a way that's based on the model. And so in principle, even subtle but consistent upregulation in certain phosphosites, for example, when we put the ligand in, we should be seeing this phosphosite go up. And it's a very subtle ratio, but it's significant. So compared to the situation where you actually put in the same number of ions, this kind of ratio at this intensity is very unlikely. Now, in practice, you're still going to do bioreps, and you're still going to go for the bigger ratios just to minimize the risk of going down an unfruitful avenue of research. But at the very least, I'm going to skip over that. At the very least, what we've been able to show is that this is a stable relationship between intensity and variance, because we did this months apart, and the variance function that we learned is basically the same. So what we can say is that an instrument, and this is an Orbi looking at eye-track data, can be calibrated what the mass spectrometrists call externally. It's sort of like an external quant calibration. You do it once, and then you know this relationship, and you can use it throughout your experiments. So that's an example of a quant challenge in mass informatics. So let me go back to the main view. So now you have the ability to call ratios that are different, and you want to represent that data. And like I said, that some of us are interested in the technology of all of this, but if you're a core facility or you're working with researchers, obviously they care about this side of things. And one of the challenges has always been, how do you represent the information? How do you make a report of all of this that brings a little bit of relevant biological context? Because the typical report is a list of peptides and a list of proteins. So I'm going to try and demonstrate a web-based tool which allows you to give a kind of a report to your collaborators, and we'll see how this works, because this is live. So I'm going to use the example graph, partially so you can redo this afterwards at your leisure. So this is a subset of the proteins in this experiment that were found in this experiment that I just set up. there's a bunch of things that you can do here, which you can also do in genomic type websites, right? So one obvious thing to do, first of all, and see which pathways we've over-enriched in, right? So focal adhesion, who are the various players? So let's take focal adhesion and actin regulation. Let's highlight those for a second to ourselves. These are sorts of moves that you can make if your data is microarray based or genomic based, right? Because you'll notice the representation here is gene centric. We're representing things as genes. I should point out we're making here a network based on known protein-protein interactions, and I'll show you in a little bit how to use this. Basically, if there's an edge between two genes, it's because we know that there's a known protein-protein interaction between them, and the color represents the source of that information. And I'll show you how to mess with that in a second. So the other thing that you can do, which you can also do for microarray data in some sites out there, is you can look at the ratios and color code based on that. Now, you'll recall one mutant we labeled with iTrack 116, the other was 117. So we can look now just at differences in the two constitutively active mutants and look at differences in the phosphocyte patterns. Two things to note here. One, because of the level of the display, I have to average over trends over all the phosphocytes that are found in these. Most of these have just one p-tire in them, so this really is a one-to-one correspondence with the underlying p-tire. But basically, and this is a very crude color coding, so these are significant ratios. Also, I forgot to mention, just in case it wasn't clear, the labels in iTrack are named after the masses of the reporter ions. So 114 is the mass that you will see for the 114 labeled sample. And so this tool, one area where we could improve is instead of these anonymous 116, 117, there should be a way to represent as a string what was the actual experiment. But if you upload the data, you typically know. But that's an area where we could improve a little bit. At any rate, all of what I've shown you up till now is relatively, it has an equivalent in other genomic tools. Before I show you something proteomic specific, I just want to do one more step, because even some genomic tools don't do this particularly well. We can, go back to what we were looking at, select, say, things that we see going up. I'm going to save these. So I'm going to start making my own little annotation of this, which is not canonical, it's just based on the experiments. So let me just collect these guys. I mean, obviously, your experiment will drive this completely differently. So you can start to collect some information that you learned from the topology and from the quantitative data. Another thing that you can do is let's do a little experiment, because I want to show you a feature. I'm going to take a bunch of these guys in a second and delete them. But I want to remember who I deleted. So here are the guys that I'm about to delete. I'm going to go to Edit, delete them. And as a matter of fact, I'm going to delete all edges, because I want to demonstrate a tool which tries to make sense of a partial set of proteins. So I'm going to select all the edges, delete them, relay out the data. So imagine that you're getting, you do a repeat experiment, and you've got a subset of the data that you were seeing before. And you would like to know, how do I make sense of this as a sort of on-the-fly pathway creation, and also what other proteins are implied by the proteins that I'm seeing here. And so if you go to topology, there's a tool called Steiner Tree, and this could be made arbitrarily complex, and it's an area of research, but I'm just demonstrating, just to give you an example. So what we're going to do here is we're going to try and connect these guys up together, and this is our data sources. And you are free to select different data sources and rank them differently. This affects the coloring. So for example, if you think protein-protein interactions from the HPRD data resource are the most trustworthy to you, when we make the network, you want the system to use those first and color that way first, and try and connect things later with other resources in this ranking. So I'm just showing you how you can select data resources and how you can affect the coloring scheme on that network. But we're going to stick with the default again, so you can redo this if you like. And so what the system is going to try and do is it's going to try and connect things with the database of facts as they exist right now. There will be a need to bring in extra proteins to bring all of this together. And a fun game you can play is to ask how many of the things that we cut out before did we bring back? So we brought back 3. In other words, in order to connect this data, it made sense to add these 5 proteins, 3 of which were what we cut out. So that's sort of a fun little observation. All of this is just in the realm of an interesting observation. The statistics for this stuff is still in its infancy, and also our understanding of the network of protein-protein interactions is, it's really premature to do this sort of thing with the databases that we have. But it's nice that it's in principle technically possible. Yeah, so all you need also to run this is a browser that runs Flash. I forgot to mention that, a recent browser. So all of this is happening in a browser. Sometimes people assume that I'm running a desktop-based app for this. I forgot to mention that earlier. So even what I just showed could be done in principle from a genomic perspective, because protein-protein interactions can be coded to the protein level. But I want to show you some aspects of the data that are truly mass spec and proteomics oriented. So if we go to, for example, this gene and underlying protein, we're claiming here that the 117 reporter ion is much greater than the 116, meaning this is a phosphosite that is much more upregulated in the D835I mutant versus the ITD mutant in FLT3 receptor. So I would like to look at that more carefully. One of the things that you can do, first of all, is look at the underlying sequence. And the window is kind of busy, but I happen to know there's only one phosphopeptide detected for this protein, which is this one. And you can see it makes sense that there's tyrosines here because we've enriched for phosphotyrosines. If I click on it, so if you're doing a classical proteomics experiment that's not phosphotyrosine, you will have typically a lot more coverage of your protein, and what this will show is a kind of build. In black will be the overall sequence, and in white will be the various peptides that you find. But so this is a little bit reduced in the sense that there's only one phosphopeptide. I'm just explaining what the nature of this component of the interface is. But basically, if you click on any of those white lines, you will be able to then look at the underlying spectrum and basically take a look at the call and see if it makes sense to you. And I just want you to note again this URL-based approach, because the idea is that this is a resource that can be extended programmatically through the web. What you're doing is you're saying, go to a website, under files, pick a file, go to the scan at this time, and match this pattern to it. This means an I-track on the N-term and a phospho on this tyrosine. You can ask it to make an alternative interpretation, and presumably we're losing some ion calls. So this is obviously, we have enough information in the spectrum to perfectly localize the phosphosite here. So one very important thing that is aspect of the data that is currently not usually supported Actually, let me go to that later. Recall earlier I said we were doing one scan for the ID and another one for the quantitation in iTrack. And so if you go to the next scan, this scan is acquired, you see that there's not a lot of signal where the peaks are that you use for ID, but all of the signal is here in the iTrack region. And as I promised earlier, the 117 peak is much greater than the 116 peak. And this is what I wanted to show you, that you can browse this data. Let's say you're reviewing a paper and you want to second guess the author. You can go into the spectrum and take a look in detail. You can pull out a text representation, so you have actually all the numbers, everything you need to look very carefully at the data. One aspect which most data resources that allow you to look at raw data don't currently do is allow you to interactively look at the MS1 data. This is very important for quantitative work and for looking at whether there's some impurity in the data. You want to look at the precursor ion. And again, I'm sorry if I'm getting into mass spec specific stuff, but for people who do this, it's currently very difficult to communicate that level of data short of giving people the raw file. And I just want to show you here that I can make what's called an extracted ion chromatogram of that peptide interactively, and you can see where it was MS-MS'd and what the overall intensity of the peak is. So for example, if you're doing label-free quantitation, this is where you would get your information about how much of the peptide there is. So again, the idea is to do these RESTful web service type system where, for example, here I'm saying, please make an XIC from this time to this time, from this mass to this mass range. And if you change this to, I don't know, 70 to 71, again, it does the right thing. If you want this in some other format, it does the right thing. I think it might do a little PDF for you if you want it. So it's a resource that will make the data in any form you want. And it's accessing the raw file. So when you share data this way, you're sharing everything. And we need a way to do that in our field because, well, I'll get to that maybe in the overview slide. This is a tool for both showing the biological context of your experiment, but allowing your customer or your collaborator to dig deep into the lowest level of the data. In the microarray world, this would be equivalent of letting them see the image and how you call the spots and stuff. Most won't care, but especially for high-level journals, there's a need right now for a way to allow reviewers to nitpick. So this is a good tool for that. So let me go back to the presentation. Maybe, so I don't know how deep we're into this. I might have a little bit of time to talk a little bit about the technological underpinnings that make this possible. Actually, just in the interest of being incredibly scattered, let me just say all of this is about peptides, but when you're talking mass informatics, you're obviously talking about people who want to do metabolomics or small molecules. And I just want to point out that there are tools in the toolkit here to do with identifying small molecules from EI spectra. And if anybody has an interest in that, talk to me later because this is a really fun area and this is a new take on that. If you're trying to identify small molecules and you you're giving up on being able to get an exact structure, sometimes you can get a sort of vague annotation or broader annotation. And this tool tries to do that, and I don't have time to go into it, but mass informatics needs to encompass all of these with equal amounts of support. Okay, so let's talk a little bit about underlying technological support. So this should jump to this slide. One of the reasons that our field doesn't get as much what you might call mind share in terms of people who want to get into the informatics of it as other topics, is that the data sets and the data types are complex. So what typically happens is you digest a sample, you run through the instrument, you get some binary formats from the vendor, you extract stuff out, you run it through some search, some identification engines, they give you back peptides and proteins, and the bioinformatics starts at that point. And so we don't have enough people looking at the underlying raw data, and it's limiting us a little bit as a topic. And so one of the things that we've been trying to do is to make tools that bring you a little bit more upstream in the data flow and allow people to look at the raw data. So I said you're getting formats from various vendors. The typical move when you're doing something like that is to say, okay, first I will extract out into some common, agreed upon, non-platform specific standard. And there's a caveat here that this can be quite tricky to do, and I just want to show you how I think as a field we've taken a slightly wrong turn in that department. So once upon a time people were measuring mRNA, they had equipment to do that, it spat out some sort of table, you could do statistics on it, and the output could be stored in a cross-platform standard called an XML file. And by analogy, I think we thought as a field we should do something similar. The difference is that our raw output from our instrumentation is still such that we don't know how to extract everything out of it, and we don't have a finite set of answers. There's a lot more in there that needs to be looked at again and again and again. So what we're doing is a lot of re-slicing the data. And the format choices, which are text-based and XML-based, don't allow you to do this efficiently. So the way we share data hinders our ability to then access it efficiently in this use case mode, when you're actually trying to slice and dice things and do computation. So in that sense, we are actually a lot more analogous to radio astronomy or something, where they have a lot of data. When they're collecting, they don't know the nature of the objects that are out there. So they need a format, they need strategies which allow you to re-look at the data in the original form and re-slice and dice it. So a while back, we published this observation and suggested to not throw out the vendor formats, but rather wrap them in a way to access the data more easily. And actually, if you really squint in here, we said XML, HDF, or API. HDF refers to a format that's actually used a lot in the satellite world, in just large-scale physics. It's called the HDF5 standard. It's a platform-independent but efficient format. And the reason I'm pointing this out is that there was a recent publication that suggested an alternative coding of the XML data in this HDF5 format. So we're seeing the field follow this trend towards not just trying to share data, but trying to share it in a way that's efficient to use. Today I'm just going to show you the end game from our perspective, which is to access the original raw data. And I'm sorry that we're getting into this sort of nitty-gritty stuff, but we are not yet, in proteomics, we are not mass spec-based proteomics. We're not nearly as advanced as in the microarray world, where you can go to a standard resource and things are really worked out. And so you're seeing sort of this in mid game. So our strategy is we will take a very easy to use language like Python, and we will wrap all of these formats in a lowest common denominator API that will allow people to look at the data efficiently. So I'm thinking, for example, if you're a core facility and you have a new kind of assay and the vendor software doesn't support your assay, you can write a script with this API, for example, to get out the data that you need for your particular new kind of label or something like that. And so when you're doing this, you need to look for common denominators across the vendors. So for example, all the vendors call their scans, this is just an example of the complexity you have to deal with, name their scans differently. Thermo names their scans 1, 2, 3, 4, 5. ABI names it a more complicated way with experiment and cycle number. And so we reached for a common denominator, which is acquisition time. So all of the APIs of all of the vendors know how to get a scan based on when it was acquired. So that's what we wrap, and that's the kind of homework you have to take on if you want to wrap around a common standard. But this is done. And so, like I said, the end goal here is to do more scriptable mass spectrometry. And one of our sources of inspiration is in the one laptop per child world. For the advanced kids, there's a mode where you can script in Python to actually script in a lot more under the hood of your laptop. And we want to do something similar for the instrument. Right now, we're doing it for the data files of the instrument. But our goal is to actually take the scripting approach all the way to the acquisition phase. And there's good reason to think that that's possible. So we want to make this stuff more accessible, more scriptable, so we can try new things. We need more sites experimenting outside of the standard operating procedures. So we made the MZ API, then we made a little desktop tool called Multipliers, which allows you to open these and do various things that one typically does, download mascot results, overlay them with precursor information, and basically then make various software artifacts that you can share. And I'm going to try and show you a few of these very quickly. You can share data in spreadsheets that you've made with Multipliers. You can share them in something called an MZ results file, which is, supports larger sharing with more data. And then in the limit, and that's sort of where my focus is right now, is to do this over the web so you can share, in principle, an infinite amount of data. And again, the astronomy field is ahead of us in that they've recognized for a long time that we need a worldwide API for accessing everything that we're doing in discovery mode that we're collecting like this. So they floated this idea of a worldwide telescope and actually implemented it, and right now you can dig into images from anywhere with just a browser. It's good for introducing students to the field, and we want a similar approach. It's actually, this is just to make the parallel even more explicit. It's just saying at some point you're going to have, if you're getting a massive grant to do discovery of an ohm, a slice of biology, and you're mapping out all these features, we're going to need to allocate a little bit of this to maintain a resource that allows you to actually access every aspect of it, not just the scans that we've ID'd or the scans that we like to show or all the various ways that people have done it up till now. This is actually in the air right now. This is a very good TED Talk where Tim Berners-Lee is saying, okay, I did this whole HTTP thing and it's great and we've been sharing and linking documents, but we need to share raw data. So I didn't Photoshop the raw data now, that's actually in his talk. And he's been talking about ways to make data accessible through browsers and, again, really raw. For some reason, his examples are always government stats data, but raw. I don't know, doesn't work for me, but it's another data type that needs to be shared, I guess. Just to highlight that we're in a bind, there's an understanding in our field right now that we need to fund something like that. So I just wanted to mention that. Actually, rather than go over this a little bit more, I'm going to try and show you some of these software artifacts that I'm talking about. If I can escape the presentation mode. Okay, it's not letting me escape. That's nice. Okay. Let me show you. So remember I said, okay, one of the first things we tried to enable sharing of data is spreadsheets, because everybody uses them, and you know, they're very straightforward. People know how to use them well. And so what we've added is the ability to incorporate right into the spreadsheet the spectrum. So when you're giving somebody a report, they can see the image, and because our tools look at the raw precursor information, not just what the search tool gets in terms of MS2 scans, we can also incorporate precursor information so you can check that it's pure. And also, you can look at the chromatographic peak. But you can see how this is a little bit hokey. It's canned, first of all, and secondly, it doesn't really, you have to move things around. It's not perfect. So the next thing that we did, which also scales up, this is work by James Weber in the lab, is to make this sort of SQLite-based format. This should start up in a second. What you need to have installed multipliers, and then this will work if you have what's called a .mzd file, an MZ database. You'll be able to see a table somewhat like the one we just saw, except that now if I look at a spectrum and I look at an image of it, it's a more of an interactive display, and you can highlight which ions are used to make a call. You can freeze that, zoom in. You can save to PDF. I'll show you later. I have a PDF that I just made of this. I don't want to waste time on that. So this is a more interactive way to share data. It also allows you, let me show you, if, let's say you have the data lying around, so I have one of the raw files, so you're in the sort of rare situation where you could actually get the raw data underlying the publication. If you double click on that row, the tool will then launch the actual an interactive display that's actually going to hitting the raw file, and you can, now you're really getting more data. It's not just interactively looking at something stored in the database, it's actually collecting data on the fly from the raw file. And as a matter of fact, if you don't have the file with you, it will try and connect through the web using the MZ server. If you have an MZ server that's hosting this file, so now what this tool is doing is it's going to our server and collecting the data. Let me just hide the one bug that's here. There. I've just recorded that I hit a bug, so that sort of cancels itself out. There's a bug in here. Anyway, what we're doing now is over the web we're accessing the data, but it's a desktop tool, so people who like desktop tools can still access these remote resources. So I'm pulling data from the a web server and throwing it into this tool. The next step up from that is the web stuff that I just showed you, and this is the underlying technology that makes this possible, is these URLs, which then hit resources that know what to do with them, and I showed you that already, so we won't bother with that now. There's a lot of support on Pathway Palette that I showed you earlier for storing things like the networks. It's vector graphics, so you can put them in publications. And if you're using MZD, for example, remember we looked at spectra. You can basically store. A lot of people wanted PDF support, so little things like that, but it's beyond the scope of an introductory talk. So is this a good time to? Okay, so we should, I guess, stop.
Any questions?
I'm just curious, what is preventing the common format from the various publishers out there? And is him as someone who created a standard.
Yes.
So would he be able to help?
Well, so there is a standards body, and it's come up with a format called MZML, which is very useful in that it is a standard. From a practical perspective, it has issues because it causes an explosion in file sizes. It's not indexed in a way that allows you to make extract it, to slice and dice in the sort of ways that you might need to if you're looking at the data, especially quantitatively. But these standards are coming about how to share the actual data as associated with the publication. There's a very good resource called Tranche, where the strategy there is it's sort of like a P2P data sharing system where you upload into this shared drive data sets that you want people to be able to download. It does some clever things like when you submit a paper for publication and you upload the data onto it, it gives you what's called a hash key, which prevents anybody from changing the data. So you can rerun the hash and show that the data that you submitted prior to publication is the data that you're downloading, things like that. So people have tried to get to this point where we can share every aspect of the data. But for example, Tranche has had some technical difficulties and is not currently funded. So it's sort of running in a little bit of a Mad Max mode where we're just sort of maintaining it. And it allows very basic capability in terms of information about the data, which is basically, here's the experiment and publication this is associated with, and here's the raw data. But it doesn't do much more than that, and it certainly doesn't allow you to interactively browse the data. But there are people working on this. We just, it's still jello on some level. I don't want to give the impression that there's nothing. Pride is another very good resource out there that's trying to come up with ways to look at the data. They have an interactive Java-based viewer. I'm waiting, especially on the capability to make extracted on chromatograms for people who label free quantitation. So we're now in the nitty-gritty, but it is starting to happen. I shouldn't.
So if a mass spec lab would want to install MZ server, so how much of a commitment is that? Is it really easy, or do you have to work on it for weeks?
So what you do is you go to the MZ server website. Here, let me just to situate this. There is a let me pull this up. We don't actually want the spectrum, we want the site. If you go here, basically what you need to do is you download one zip of various little files, you download multipliers, and you download, so you either have an instrument, in which case you're done. If you don't have an instrument, because remember, one of the things we're trying to do is get people who don't acquire data looking at it. So Thermo, which is the vendor that has been most ahead of this game of trying to share the raw data, has made a DLL that you need to download from here. You download it, and then you basically just run. It's packaged as an MZ script. Let me show you.
So you can pretty much be up and running in a few hours.
Yes, I didn't mention, when you install multipliers, if you have a little text file, I just, let me show you this really quickly. This little example.mz, it's what's called an MZ script. It's a very simple script. In this case, it's just opening this raw file and making an XIC. If you have multipliers installed, it will just run. So it's going to there. It just opened a file, make an XIC, and printed out the value of the maximum. The maximum was min51. That was the intensity. The point is that the MZ server is just another MZ script. You just double click and it opens a server and it shares data. The data is shared from a directory. You just drag and drop a raw file in there, and you can at least browse it from your own intranet. You probably don't want to put this up there willy-nilly. You want to first share data internally through browsers before you share it to the outside. But yeah, I obscured the point that this is relatively straightforward to set up.
Thank
Sorry
you.
about that.
Thanks.
Can you distribute data with this too? Or is it just for viewing in like a browser?
So the idea is that, so one mode that's right now, a functionality that is disabled or not implemented is when you're looking at a raw file, if you open here, so let's say we're looking at some scan, right? If we go up a level, if you select this, it will make a report of every scan in the file, which will take a bit of time. There you go. So these are all of the scans, links to all of the scans. If you go up one more level, what you'd expect to happen is that this will cause the raw file to be downloaded. This is what you want. And right now, what it does is it makes a total ion chromatogram. The reason is, if we made that possible, people would start downloading from our demo server raw data, and the bandwidth would be problematic. But this is also kind of sweet in that you can basically look at the entirety of the file and click to go to any of the MSMSs. Right? This is the file we were seeing earlier. So in the long run, you shouldn't need the entire raw file. The next step here is, right now you can pull spectra or XICs or various analyses of the data. We want to make more and more of the computation happen at the server. So higher level algorithms, dot product, searches of similar spectra, you should be able to tell the server to do things on your behalf. So the challenge is how to do that securely and not crash the machine. So, but the short answer is no. you can't download the raw file.
So I'm interested in the potential research use of this data transfer, something synthetic. So you have phosphorylated proteins, and they of course have different ligands that they phosphorylate, different proteins that they phosphorylate, say esrin or mollicin, to that same receptor. Could you use this form of data transfer to understand what you might be looking for in, say, a phosphatase or?
So one use case that I want to make a demo for, I think let's do a little bit of science fiction, is you're looking at the data from pathway palette, you look at a pathway, maybe if I can, here. If you're looking at a pathway, here, you can look at some of the nodes that you didn't see. So you can expand out to proteins that you didn't see. Then you would want to get their sequence, predict the mass, go to the raw file, and make an XIC to see whether the peptide was there. And so you saw how you can match the XIC, and so it would be pretty straightforward to make a tool which uses this to do some context research, then makes the relevant URL and hits the raw file if the raw file is exposed this way. So I think that's sort of where this is going a little bit. You're right.
Sorry, we're going to have to stop the next meeting and start it. Sorry about that. Yeah.